Hi everyone, in this narrated PowerPoint, I am going to give you some background information on cystic fibrosis, and we'll discuss the Watkins paper as a big overview where they were able to make some progress in mapping disease genes using pedigrees. And then finally, I'll give you some notes and some information about the next steps after mapping, which is to clone disease genes. So our upcoming topics are going to be moving on from mapping to cloning human disease genes. So the assignment due for Thursday, March 26th, is based on the reading guide that is now posted in Blackboard. And the big assignment that's coming up for the following Tuesday, March 31st, is your first draft of your research paper, parts 1 to 3. In the red box at the top of this slide, I'm reminding you that this is posted as an assignment in the Module 3 Assignments tab. So all of the information on how to write the draft and what I expect to see in the draft is posted there. But if you have any questions, please email or attend one of our WebEx sessions to ask. And I want to remind you that you really need to start finding articles for parts 1 to 3 of your research paper now. Some of these articles you will need to get using interlibrary loan, and that can take a day or two. So just keep in mind that you're going to need to cite all of the information you include in your paper. You cannot cite any websites. And so if you have any troubles on getting started, finding papers, or anything regarding the research paper draft, please let me know. Okay, so back to kind of getting started on cystic fibrosis. And this is the disease, again, that I've chosen to sort of model what I'm expecting you to include in your research paper. So on this slide, I'm showing you some excerpts from a 1991 review article. And this is written by Francis Collins, and his lab was one of the main labs that was successful in cloning the cystic fibrosis gene. This review article was written 2 years after they published the cloning of the gene. So first we want to understand a little bit of background on this disease, why we should be interested in it. And it is an autosomal recessive disease, and the frequency is fairly high, 1 in 2,000 live births in North America. The symptoms are chronic pulmonary disorder, pancreatic enzyme insufficiency, and because it's an exocrine disorder, it affects several glands. Okay, so that's a little brief background on cystic fibrosis. We'll be coming back to the symptoms, diagnosis, and treatment of the disease in some later lectures. So on the right-hand side of this slide, I have posted a little bit of the introduction to this review article. And the name of the review article is Of Needles and Haystacks: How to Find Human Disease Genes by Positional Cloning. And the very first sentence of this paragraph tells us that it took almost 10 years to hunt and search for the cystic fibrosis gene. So this was a really large undertaking, and it took the efforts of multiple labs working together to eventually be successful. So in the first red box on the right, I have kind of set aside where Dr. Collins says that the first step in this cloning of this type is going to be mapping the gene to a particular human chromosome using linkage analysis. And so that's where we use the pedigrees, and that's why we're looking at that the Watkins paper, where they do some of the mapping that was really important and led to the eventual cloning of this. So this type of cloning is called positional cloning. Okay, so this means that you really don't have very much information at all about the location of the gene within the genome. And Dr. Collins goes on to say that this type of positional cloning is a labor-intensive process because 98 to 99% of the markers tested are going to be either on the wrong chromosome or really far away from the disease gene. So they're not going to show any linkage. So that means that most of the time, as you are checking different markers to see if they are linked with your human disease gene, you're going to get negative data, right? So that means really low LOD scores or negative LOD scores. And that can be really discouraging. Okay. So here, Dr. Collins included a figure, which I just like because of his analogy that positional cloning is like a needle in the haystack activity, right? You're looking for a gene that's within 3 billion nucleotides in the human genome. And so it's difficult for sure. Okay, so as I mentioned, the Watkins paper was one of several mapping studies that helped lead to the successful cloning of the cystic fibrosis gene. So the Watkins paper was published in 1986, and the papers that we're going to be talking about in the next narrated PowerPoint were published in 1989. So this paper was still 3 years before they eventually cloned the gene. Okay, assignment 11 was based on the Watkins paper, and just to remind you, what they knew prior to the beginning of the study was that the gene underlying cystic fibrosis was on chromosome 7, so other mapping studies had already been done, and they knew 3 genetic markers that were closely linked to cystic fibrosis. Those markers are MET, PJ3.11, and 7C22. Okay, so they already had some good genetic markers that were linked with cystic fibrosis. So what questions remained at the start of this study? Well, there was still some question as to whether or not cystic fibrosis disease is associated with only one gene, or could it be associated with multiple genes? And that would be locus heterogeneity. And they also were still grappling with the precise order of those different markers and the cystic fibrosis gene along chromosome 7. So in this study, they analyzed 2 large pedigrees and 17 nuclear families, and they were looking for linkage between these markers and cystic fibrosis. So they used DNA probes for each of the 3 genetic markers, and they performed southern blots. And then for one of the markers, they were actually able to look at enzyme data. And again, they're looking for correlations. Does the marker, the presence of the marker, basically a band on the southern blot, or positive enzymatic activity, are these markers linked with cystic fibrosis? And then they were looking for recombinants so they could calculate LOD scores that would inform them as to whether or not linkage occurred. So the results in the Watkins paper are all in Table 1, and that was on page 740. And in this table, they reported the LOD scores between cystic fibrosis and each of those 4 markers. The LOD scores were for 9 different recombination rates, and you can evaluate the highest LOD score in each row, and those are going to be in the first or leftmost column where theta is 0.00. Okay, so that was part of your assignment, was to kind of take a look at that table and to look and see what LOD scores they obtained, to see which markers did have significant linkage and to see if any did not. By the end of this study, which was a fairly small study, was the location of cystic fibrosis narrowed down very much? Well, somewhat. They had narrowed it down at the end of the study to 1.5 million base pairs between the marker MET and J3.11. Okay, so now you can kind of see why it took another 3 years before the gene was cloned, because 1.5 million base pairs is still a lot of the genome, a lot of chromosome 7 to sort through. Okay, so positional cloning, that's where we want to spend the rest of our time today, talking about what this approach is and how it helps us to clone human disease genes. This is one of several different strategies that can identify human disease genes, and we'll be discussing some of the different strategies in a different lecture. So today we're focused just on positional cloning, and this is where the approximate chromosomal location is used to eventually identify the human disease genes, and as we saw from Dr. Collins' review article, it's very difficult work. There are 3 basic steps in positional cloning. Step 1 is to define the candidate region as tightly as possible. And so once you have a region of about 10 megabases, you can then start to try to collect as many families and pedigrees as possible and determine these maps of these polymorphic markers. So that's the genetic mapping that occurred in the Watkins paper. Linkage disequilibrium can sometimes help generate high-resolution maps for some diseases, and this did help for cystic fibrosis, but not all diseases show this. And remember, linkage disequilibrium can really probably most simply be thought of as just linkage between a marker and a disease. Okay, so after a lot of mapping efforts, step 2 is to try to identify genes within that smaller candidate region. And you can identify genes based on having evolutionary conservation, right? So if this chunk of DNA is really encoding an important gene, we would expect to see that it is conserved in closely related species. Another part of the process of considering whether a candidate might be correct or not is transcript analysis, and then there's some other criteria on slide 10 that we'll go through. Okay, so the evolutionary conservation. As I mentioned, critical genes are conserved. So what people do for this is southern blot analysis. So they collect DNA from various species, they run it on an agarose gel, and then they use a probe for the candidate gene, and they ask whether or not the DNA probe cross-hybridizes. So even if it's a different species, if they're closely related species, we would expect that the probe would be able to bind to the gene if it exists in those other species. So basically, if you see bands on a southern blot, that's going to be telling you that this particular chunk of DNA is in fact conserved. Transcript analysis would entail the idea of if you've got the right sequence of DNA for your gene, you would expect it to be transcribed into RNA. So in this case, you could do a northern blot where you're running RNA on an agarose gel, and then you're going to use a probe to see if it binds, which would be evidence that the gene is in fact made into RNA in particular tissues that you would expect. So for example, cystic fibrosis is known to affect lung tissue, so that suggests it's going to function in the lungs. So that might be some of the tissue that you would use to extract RNA from and run on your northern blot. We'll see an example of this when we look at the Rorden paper, figure 3, in a future PowerPoint video. Okay, so you can basically look through these different candidate genes you have and see if you can narrow it down further to perhaps just one gene that meets all the criteria. And at that point, you can then screen cDNA libraries to clone the gene and get the sequence of the gene. So positional cloning has been pretty successful. You can see that in 1986, the gene for Duchenne muscular dystrophy, the gene is called DMD, was cloned. Cystic fibrosis, 1989, as we've talked about, that's the CFTR gene. And you can see several other diseases are listed here, including some that students in our class have picked as topics. Okay, so I mentioned briefly that linkage disequilibrium was a tool that was used in positional cloning, especially for the cystic fibrosis gene. So several DNA markers were successfully used to map cystic fibrosis to the region of chromosome 7 called Q31, Q32. So that's just a particular spot within the Q arm. And the flanking markers were eventually narrowed down to MET and D7S8. So remember, MET is one of the ones that we saw in the Watkins paper. After the location was narrowed down, a method called chromosome walking and chromosome jumping was used, and we're not going to talk about those methods in great detail, but that helped them narrow down the region even further. And then linkage disequilibrium helped provide valuable clues to help them decide which markers were really closely linked and where they should focus their attention. And as I said, library screening is eventually done to clone the gene to get the sequence. Okay, so at the end of the day, what we're going to see in our future PowerPoint videos when we look at the Rorden paper very carefully is that they were looking at 4 different candidate genes, and in the red box at the top of this slide, they say the first 3 genes failed to pass the genetic test, but the 4th one eventually proved to be correct. And then they talk about the fact that on a northern blot, they saw the gene encoded an mRNA that was 6.5 kilobases, and they said that they saw expression in tissues where cystic fibrosis is evident, like the pancreas and the lung and sweat glands. They go on to say that they also were able to validate and prove they had the correct gene because in 70% of patients with cystic fibrosis, there was a particular mutation. It was a deletion of phenylalanine, 1 amino acid, at position 508. So basically a 3 base pair deletion. So it was just the loss of this 1 amino acid that led to all of the symptoms and phenotypes of cystic fibrosis. So obviously this is not all of the patients, it's just 70% of them, but it's quite quite remarkable, right? That if 70% of the patients have a particular deletion, it seems that this gene is very important and is the causal gene. And in particular, that phenylalanine amino acid must be playing a particular role in how that protein functions. And we'll talk about that next time.